Cool. Okay, so we'll see how far we get today, because I basically, it's a bit of a chunky lesson, because I want to start introducing our next internal, which is on quantitative analysis. Sounds very similar to what we just did, because the word that we were thinking about last topic was qualitative. This one is quantitative, so I want to kind of go through the purpose of it, and then also start showing you guys the titration equipment, which is how we're going to be able to quantify things. For your do now, that was just a refresher on some of these terms, and it's going to tie into what we're going to be doing for this topic: solute, solvent, and concentration. Solute is the solid substance or maybe a liquid that you're mixing into something. The way I like to remember is that you're soluting as you're jumping into the pool. Say, for example, salt that you add to water. The thing that's going to be the liquid that dissolves the substance, or the solute, is going to be the solvent, and it's venting for this solute to hurry up and dissolve. Was that clever?
Yeah.
Okay. I thought it was kind of silly, but it helps me remember. And then concentrations is talking about the amount of the solute that you have in the solvent. So it could be very concentrated and very strong, such as like a strong coffee, or maybe you add some water to dilute it so that way it's not as powerful. So that's kind of some key terms that we're going to apply to this topic. So let me give you an overview of the lesson plan. Come on, you can do it, computer. There we go. I should also find my clicker so that way I can actually walk around. So what we are thinking about first is how we connect the internal that you guys just did to the next internal. I think it's good to see that, because that way you understand kind of some really important science skills. Then what I want you guys to do is have a chance to kind of think of a real world example of when we do quantitative analysis. I'll give you guys some information then on the different techniques we use to figure out concentrations. I will talk about the assessment as well today, and then I want to give you guys a chance to explore the titration equipment. There's this really cool thing that some of you guys might have met Mr. Hall, he's a student teacher. He found this cool website that has online worksheets. So I've selected one of those, and then if we have time, I'll start bringing out the equipment so you guys can have a tinker and kind of look at what you're going to be working with over the next couple weeks. I've also decided to start giving you guys homework just because I'm kind of worried about the pace that we're moving, and I really want to encourage you guys to work outside of class. With the homework, what I'm going to do is give you stuff that's related to the internal, but I'm also going to give you some guidance on what you should be doing in preparation for the external as well, because I want to make sure you guys kind of feel that sense of urgency, because that's basically what we're kind of feeling, and really utilize those Fridays that you're going to be given to do your independent kind of work. Are we good so far? So for today, I would say that you should be making sure you have page 12 and 13 done as your kind of homework for tonight, because that ties into our titration stuff. Cool. All right, so let me pass out the checklist for this new topic. I put it on pink paper, and what we're just doing for today is defining those key words for our titration topic. You'll see there's a little meme that I put on there. This meme is a very accurate description of how you're going to be feeling when you are doing titration, because it does get quite frustrating at times. Can you guys make sure you write your names on your side pads as well? I have Sharpies up here. That way, if anything goes missing, I can make sure it gets back to you. So that's all one side. Cool. All right. Let us keep talking. So, this PowerPoint is online, and I printed some notes for you guys later to highlight kind of the key things, but this I haven't printed off because I didn't feel like it was that urgent, and I wanted to save some paper. So, we just finished up Chem 2.2, which is qualitative analysis. So basically, we were asking the question, what do I have? What's in this liquid? What is present? So after we have an idea of what is there, our next question is going to be, how much is there? Two very key questions in any sort of scientific research. If you remember when we were talking about Chem 2.2, you had your little essay component, and if you're trying to get the excellence, you need to really talk about dosage and concentration, because that can influence if it's a beneficial thing or a toxic thing. The thing I like to remember that my supervisor always said to me is that everything is toxic, it's just a matter of dose. And so that's why that concentration information is so important, because at a low amount, such as paracetamol, it's helpful, it manages pain relief, but if you take too much paracetamol, you can end up in the hospital with liver failure. So that's why that dosage is important. And in fact, even water could kill you. If you drink lots of water and you don't go wee, your brain will swell. And in the States, they actually did that competition, and someone died from it. I told you guys about
for a wee.
Yeah, and it was for a wee. Like, hold your wee for a wee was literally the competition. And people were calling in and saying, you need to end this competition. She's clearly, her brain's swelling. I am a nurse. Do not do this. This is so dangerous. And she ended up dying. I'm not sure if they won any lawsuit against it, because you signed a disclaimer, not disclaimer, like a liability waiver, but I don't know if that, like, had any effect. Anyway, quantitative analysis, like I said, is about quantity. So asking the question, how much is there? So once I know what's there, I want to know how much there is. So for you guys to start thinking about the importance of quantitative analysis, and also comparing, contrasting qualitative and quantitative analysis, I put a link on Google Classroom from a Fairgo article looking at beef mince in stores and the quality of that beef mince. So you guys can work together in groups, you can work individually, but I want you to have a read of the article, and I want you to think about these questions. So from reading that article, what's an example of qualitative analysis? What's the health impact of that substance? What's an example of quantitative analysis? Why would people care about the results of the quantitative analysis? And how does this affect consumer behavior?
What do we write out?
You don't need to write it down. I just want you to discuss it with the people next to you. Yeah, don't worry about writing any of the answers down. It's just to help explore the real world impact of the science you're learning. Cool? All right, I'll walk around and help. Oh, cool.
Can it speak?
All right, cool. So, I really like this article because it shows you the real world implications of the stuff that we talk about in science, or in particular chemistry. So, first off, we're thinking about the qualitative analysis. So in that case, we're just trying to identify something. So what in the article were they trying to identify? Not the fat, the sulfites, yes. So the sulfites are used as a preservative thing. You find it in some food and you find it in some beverages as well. It's just to extend the life of items. Yeah. But it is something important that we need to know if it's there or not, because some people have allergic reactions to the sulfites. And that's really dangerous, because when you think about it, someone's going to eat something and then allergic reaction, and allergic reactions can kill you. So that is concerning that is something that they tested for and they could find in some of these meat products. So what was the quantitative analysis of this article? What were they trying to quantify? You want to take a stab at it?
Mince.
Yeah, the mince, and what particular about the mince? Because you said it, you started saying it earlier. You see it on the packets when you buy it.
What is this?
The fat content, yeah. So they were trying to quantify the fat content versus the meat content. And you guys notice that when you're at the supermarket, when you're picking out the mince, that some mince is obviously higher quality than other mince, and you might pick certain ones based on price or based off the quality of it. So that's really concerning. Do you think people would care about the percentage of the fat in the mince?
If they have heart problems and stuff.
Yeah, they could have heart problems, they could have other dietary requirements, they could be trying to cut back. So do you think this would affect how consumers would shop? Yeah. So this is really important for us to think about. What this is actually referring to is something known as, once my computer works, quality control. So, quality control is the example that we are thinking about. This is actually a whole area of science. You can get employed as a scientist to do just quality control checks on products before they go to the consumer. I actually did some of that when I was for one of my summer jobs. It's really kind of neat that you need to check to make sure it works before you send it out. Otherwise, you're not guaranteeing your promises. So let's think. Huh? In one house, yes. So some other examples of what we might need to use quantitative analysis. So if we want to know quantity. So this first one here, what is that one about? Do you think? The insulin pump and making sure you have the right amount of insulin throughout the day. So this is another thing that we would want to quantify. So in a medical perspective, you could use it from your day-to-day health. Some people you might know actually have diabetes, and they have an insulin pump to help regulate their sugar levels. But Amy Winehouse, why do you think she's up there?
She died of heroin.
She died, and when they did the toxicology report, she died of alcohol poisoning. It was probably a mixture of drugs, but you can see how you can identify what's in her system, but the quantity is important because you need to know, is it the overdose amount? So again, that quantitative analysis is really important. All right, this one ties to what we were thinking about in identification. You can see they're testing the river. Why do you think they're testing the river?
For
any poisons, for chlorides, for things like that. And again, we're thinking about that life force of that river. So we need to figure out what's in the river and how much. And you guys were thinking about that with the last topic, because in the last topic, you were basically just identifying what was there. And in this topic, I want us to now figure out, okay, now that we know what's there, how much is it, and is it going to be a problem? Cool? So I just want to see you guys see those real world examples. There's lots of different areas we use this, and it goes all the way from health to cool things like forensic science to things like making sure we take care of our environment. All right. So this is what we actually do for concentration analysis in the fancy sense. This is what I did my master's in. So for my master's, I did analytical chemistry to figure out how much I had of something. So I was pretty interested in something called glutathione, which is an antioxidant. It just helps kind of mop up things that could be harmful in your body. And what I did was I used this HPLC machine. So this is a really fancy machine. I took what I knew as known concentrations, I created a line, and then I created an equation. So when I had an unknown sample, if I knew the amount of absorption, I could plug it into this equation, and it backtracks and tells me concentration. So I'm using some sort of mathematical method to figure out my unknown. Now you guys won't be doing this because that machine's about $100,000, and we're going to do something that's kind of more old school budget. So here's our budget version. I'm going to pass out the notes now because this is something that I do want to show you guys. I figured this might actually be helpful for you guys to just know. You can jot down some things I'm saying. But here's our budget way of doing something. Yep. What's in here? All right. So here is the budget way of us doing this. Because we can't afford HPLC machines, and I don't think you'd want to be on an HPLC machine. I've broken it multiple times, and it's very terrifying to break something that's $100,000. Good news is it was easy to fix, but it's not something I think you guys want to be responsible for. All right. So, get my little thingamabobby. Yeah. All right. All right. So let me just run through some ideas, and then you guys will have a chance just to tinker with this as a concept. So the budget way we do this is by something known as stoichiometry. We were supposed to talk about this beforehand because we would have finished the structure and bonding, but we didn't finish the last section of structure and bonding, which was on these calculations. So stoichiometry is going to be something new for you. But the good news is when we go back to the external content, you then have some foundation for it. So stoichiometry is taking advantage of the fact that we have equations. So if you guys remember in our last topic, you guys were writing equations, and one of the things you need to do was balance them. We can then use this information to figure out what the unknown amount is. So for example, if I'm thinking about hydrochloric acid and I want to neutralize it with a base, when I neutralize it, I make water and I make a salt. When we are looking at this equation and we're trying to balance it, it's all a nice 1-to-1-to-1-to-1 ratio. So what that tells me is the amount of hydrochloric acid that I have is going to equal the amount of the sodium hydroxide concentration. So that way they balance out and get neutralized. Are we okay with that so far? So if I know the concentration of one thing, I can figure out the concentration of the other thing.
Oh my God.
And that's what we're going to be using as our reference point. So I want you guys to have a chance to have a little play with this. So let me kind of just walk you through what I want you to do. So I have taken out some hydrochloric acid and some sodium hydroxide. I'll get the little pipettes out as well. You guys can see ones. And you're going to be opening up the bottles, because I don't want to open up more, or I don't want to get more beakers out. And you're going to be using this pipette, and these have little measurements on the side. And in theory, if you can get the ratio between the hydrochloric acid and the sodium hydroxide equal, you should have a perfectly balanced neutral solution.
We can drink it.
No. Well, I mean, yes, you could, but no, you're not doing that. Because it's going to be salty anyway. Yeah? So, when you guys do that, you guys see on these bottles, they're both the same concentration, so it helps balance it. If they weren't the same concentration, then we might need to do twice as much as one thing to balance for the fact that something else is more concentrated. So we're keeping it easy, same concentration. The other thing that I'm going to give you is universal indicator. Have you guys come across universal indicator?
Yes.
Some yes, some no. So universal indicator is something that changes color based on if something is acidic or basic.
Yeah.
So if something is acidic, what color was it going to be?
Green.
Acidic. Red.
Red, yeah.
If something is basic, what color is it going to be?
Green.
Blue, purple, pepper. If it's neutral, what should you get?
Green.
Green. So I'm just going to let you guys have a little play to see if you can get your solution to be green. Okay? I'll get some more pipettes. Make sure you put on your goggles. And you can come over here, grab yourself a beaker, grab yourself a bottle of acid, grab yourself a bottle of base. And
Oh,真的吗？
So try to get it so it's neutral. Oh.
You're inside.
Wait, come on. Get your goggles, let's go. Whoa. All right, so just gonna pull up the notes again. How did you guys find that? Was that easy or hard?
So easy.
Some of you guys thought it was easy? Oh, it was supposed to be hard. All right. Would you guys want to do your assessment that way?
Yes.
Do you know how much you added to it?
Yes, we did 5 exit and 3 of the What's the other?
The other option is we do this with proper equipment and you do it actually a lot more accurately.
Okay.
Okay? So, some things to note is that by doing what you did with the pipettes and just kind of squeezing it into a beaker, what you were trying to do was get that ratio between the acid and base perfectly balanced so that way you had the neutral solution. But a lot of you guys, if you remember, you overshot, you undershot, you got the wrong direction, you got to go backwards. And that's not a very accurate way to measure, Ruben. Not off the top of my head, sorry. Anyway, so what we do to help you guys measure more accurately is we use some titration equipment. So there's things such as burette, and we use a glass pipette. It's kind of different. We still use a universal indicator, and we're going to use those to accurately figure out the volume of each solution. We know the concentration, we know the moles, and then we can do some calculations. So that's what we got to keep in mind with this titration equipment, and I want to start exposing you guys to that today. Does that sound good so far? Yeah, okay. So we have our flask with our solution, in this case acid. We add some universal indicator. In this case, it's going to be a colorless in a base, and then in an acid, and your burette has some base, and you add it to it, and it's going to turn pink when it's neutral, or when it starts getting towards that neutral end. So that's basically what we're going to be doing for this practical assessment. Now, let me give you guys an idea of what this internal is going to be like, because I don't want you guys to be super overwhelmed by it. It is an investigation. So some of you guys are taking biology and you're doing a biological investigation now. So it's the same kind of concept. You guys are going to be doing an experiment, and there's a write-up component to it. The good news is that this one is broken up into different pieces, and it's a lot more scaffolded than the biology one. So it's going to be hopefully easier in your eyes. So it is an internal. It is worth 4 credits, and it's broken down into 4 different parts. So there's part A, B, C, and D. There's 4 parts to it, and you're going to need to pass each part to pass the internal. And then the part that is the lowest is going to be your overall grade. So it's a lot less forgiving than the ion identification one. Are you okay so far? The good news is, what we're going to do is I'm going to teach part A, you're going to practice part A, I will assess part A, and then we're going to move on. So that way, it's not going to be, here's all this information, now do the whole assessment. So that's the other nice thing about it, is that we break it into chunks. So in the first chunk, you guys will be given some solutions. You're going to just be tinkering. You're going to do some math, and you're going to figure out a method from there. And luckily, the method that I can give you is scaffolded. After you do that, we have part B and part C. Part B and C are very similar. They have a very similar calculation. You're just working in different directions. So basically, part B, you go from 1 to the back, and then part C, you're doing the exact same thing, but you're basically working the steps in reverse. Are we okay with that so far? Part B is just a written test, just to do the calculations. Part C, very similar calculations to part B. The difference with part B is that you will be doing the titration and collecting the data yourself. The good news is, for part C, I will allow you guys to work in pairs for that. Okay? And I had to think through, and there's going to be certain sections that I'll allow you to bring in, kind of like a checklist or a cheat sheet to help support you so you can remember everything to do on the assessments. So that's part C. And then part D is kind of your evaluation and write-up. So in this case, again, it's a written component, and you're just kind of looking at all of the assessments. So I'll give you A through C back to you, and you'll be reflecting on the method you created in A. You'll also be talking about the manufacturer's plan. So the manufacturer is going to say, you have this amount of vinegar, or this amount of ethanoic acid in my vinegar, and you're going to verify, is that true? Just kind of like when we were looking at that Fair Go article, and they're asking the question, is the amount of fat they're saying is in the mince true? So you guys will be able to do that. That's the really cool thing about it. The other good news about some of this assessment is, if you're aiming for merit or achieved, it is a little bit more forgiving. So from memory, if you screw up part A, there is an opportunity in part D to kind of fix it. If you are aiming for a merit, you just need to do the calculation once properly, either B or C. If you get the calculation wrong, you can still get a merit. Excellence is going to be quite intense, because you're going to have to do everything really, really well. But don't worry, I'll walk you through it. Each one we do, I'll explain to you what the achieved, merit, and excellence requirements are. And like I said, we'll assess it after each teaching, so you don't have to retain all the information at once. Are you okay with that so far? All right. Cool. So I'm just mindful of time, because are we ending at 11? Today? Okay. There's this online worksheet, but I'm mindful I also want to show you guys the equipment. But I know I've been talking for a while. Do you guys want me to start showing you the equipment? So you guys can tinker with it? Okay, cool. We can start doing that. So let me go grab the pipette and the burette so you guys can see what that looks like. I need to grab one of these guys, which is our plant stand. And I don't know why there's two on here.
Oh, yeah.
Actually, while I'm setting up, you guys can try that online worksheet. All right, cool. And I'll put it on the screen so you guys can also see if you're a little bit further away. I'm just gonna be mindful where I'm standing. You okay? So, I just want to show you guys some of the titration equipment so you guys are aware of what you're doing and what you do it, why you do it. So this first thing here, let me actually get this removed so you guys can actually see the whole thing. So, get that guy off. This here is our clamp stand. This is what we're going to be attaching our burette with. The burette is luckily plastic, so I'm not so worried about you guys breaking it, but the last ones, those are terrifying. What you guys notice on it is that it has these little measurements on there, just like you would have as a measuring cylinder. But the thing that's different about these guys compared to a measuring cylinder is that the bottom is 50 and the top is 0. Whereas when you guys have a measuring cylinder, the top is 100 and the bottom is 0. So it's different because the liquid's obviously coming out the bottom here. Yeah, so I'm going to show you what I mean by how we're going to read it. So, I'm just putting this up. I am going to make sure the valve is closed so that way water doesn't go everywhere. I'm going to put my flask underneath it. I might need to switch this around a bit so you're going to be able to tinker. Now, the thing you have to remember with this equipment is that because we are doing titration, we're trying to accurately measure concentration. Something that's really important is that you guys have separate equipment from the normal classroom stuff. And that's just to keep it nice and clean, so you don't have any cross-contamination. The other thing that's going to be really important is that we're using something called distilled water. Have you guys heard of distilled water?
Yeah, it's water that's
It's water that's distilled. Yeah, that's a good start. Someone's going to Google it. Alright, that's a good one. Alright, do you have an answer? Purified water. So what I've done in this is that this water is purified. It's removed all the ions from it and things like that so you don't, again, have that cross-contamination. So something to remember with this equipment is you're never using any of the tap water through it.
Never.
Okay? Never.
Only distilled water.
Only distilled water. Okay? The reason why I don't want the tap water is because they have little ions, and if the little ions stay in my equipment, it could crystallize and clog my equipment. And then on top of that, it could also affect your readings. So I'm just doing things with distilled water for today, and you guys will have a chance to do this as well, just so you have a chance. Oh, I overfilled that.
Oh no.
Oh no!
Oh my god.
Release some of that.
Something's gonna come out.
Yeah, like
Alright, I'm just going to remove that to give you guys an idea. So, what you guys notice here is that I filled it from the top like I would a measuring cylinder, but instead of pouring out from the measuring cylinder, we open these little valves here, and that's going to release the water, or in this case, chemical. Now, you can obviously control that valve and how much you're releasing, and that's going to be really important, because if you want to try to get it really, really accurate, you might want to do the little drippy drop method. Yeah? So you can get it to go really, really slowly. And that's what you guys are doing when you're trying to neutralize that acid and base. You're like, ooh, just one more drop, just one more drop. And that's kind of mimicking what you're doing with this burette. So that's kind of the first thing to think about. I want to show you guys how to read that burette, but I have an image for you guys to help you see it. So, zoomed up on it because I'm mindful you guys can't see it. So, you have to remember with these burettes is that you're reading it opposite to how you would read a measuring cylinder. So measuring cylinder, you normally go read up. You're going to be reading down. So if you're reading this incorrectly, you would go, oh, that's 47. So that must be 47.1. That must be 47.2. So it must be, since it's between 47.2 and 47.1, it's 47.5 is what you would think if you were using a measuring cylinder.
46.
You guys see what I'm saying with that? So that's the incorrect reading. What else did I want to say about that? Make sure you're reading at the bottom of the meniscus whenever you're reading liquids. So the correct reading is actually going from top down. So you're actually at 46 and you're reading going 46 down in that direction. So if I was looking at it from the screen,
So look, if you 48 that's 47.
so this is 48, 49, 46. So you guys are used to looking at measuring cylinders and you're used to reading it from the bottom up. So if I had 35, it would be here.
Usually that would be 40.
Does that make sense?
Yeah.
But in this case, because the water's coming down at the bottom, you're going to be reading it from top down. So that's 46. That's 46.5. And then if I go 46.6, 7, 8, 9. So I would say it's roughly between the 8 and 9 value. So I would say that's 46.85.
So,
Do you see what I'm saying? You're reading it in a different direction. This is actually a really common mistake, and if you make this mistake, all your numbers are going to be very off,
you get it?
and you're going to get it cheat. So that's the first thing I need to tell you guys. It's different from a measuring cylinder.
I mean, I thought it was like straight on point.
Yeah, so if this line was right here, you would then say it's 47.
Yeah.
And then once it goes past that point, right there, that's 47.1. Does that make sense?
So
Is that okay so far? All right. So that's the first bit of equipment that I want to show you guys. You might have a chance. Do you want me to show you the other one now, or do you guys want to have a try of this one?
the other one.
The other one too? Okay. We got it all done, and then you guys can spend Monday. Cool. So the other thing I need to show you is a pipette. So, you guys okay? These are a little bit dusty because they haven't been used. I think people have different preferences. And I'll grab the other one as well so you guys can see choices. So, you guys will have a lot of choices on which pipette you want to use. I personally have only used these bulb ones. I've not used these ones before.
Oh my god. So
you guys would know. You know how I was talking about the pipettes that you were using? These are also known as pipettes as well. The whole thing about these pipettes is that they're meant to measure and aliquot, so to kind of give a set volume of liquid. Aliquot is just a fancy way of saying like a chunk of liquid.
allocate.
Yeah, you're allocating it.
So when you're writing a method, you just call it both.
You can call both pipettes. I've seen that plastic one known as a dropper as well, but it's the same idea, just a bigger version of it. So those pipettes I think go up to 2 mils. This one goes up to 25. So that's the only thing that's really different about them. So with these guys, there's some choices. There's this one here. These come in different sizes, but the downside is it only does 25. This one here, I actually haven't seen before. My previous one didn't have them, but they're more accurate to what you guys would be using in the lab. And this one goes to 25 as well, but you have to measure it against it. So if I wanted to get 25 milliliters, I could put this in, and this one works by a suction. So you pull it up like that. So some of you guys might like this one better. The problem with this one is that there's a lot of noise because you have to think about all the measurements on the side. The good news is it gives you that flexibility. So if you need 10 mils, 25, 20, you can do that. But you just got to be mindful about where that meniscus is and not reading it next to the plunger. So that's kind of the downside. Some people like this one. I haven't seen this one before. This is more accurate to what you'll be using in the lab at university. or if you're doing anything science-wise, and then releasing it, you just push it back down.
I thought the meniscus was over. Or your meniscus.
I'm not sure. I only heard meniscus as like
Oh, you think meniscus?
meniscus.
Oh, yeah, meniscus. That's the.
Yeah, that's the little, because you know how water has polarity and it clings to the side of the container, so it's going to cling a little bit to the side, and that's why you have a little blob on the bottom. I have mostly used this one in high school. This one here is only 25, and you're going to be filling it to the line. So some people might find that easier. When you are doing that, you have these things called bulbs, and there's lots of different versions of bulbs. This is my own personal preference, but there's two other ones you can get. I'll grab them actually so you guys can see it. Yeah, there's lots of them. So there's this one here. I'll show them on camera. There's this bulb is your first choice, if you like that. This one you just suck up. You have very little control. This one here is like a crank, so you pull the solution up that way. I don't like this one because I find that sometimes you don't get enough solution, and then you have to like pull it off, do that, and put it back on. I don't like this one. All right, and this one is probably my favorite because it allows you a lot of control. Actually, between these two are probably the two best ones. I'll take this one though, actually, because I think this one's better. So, with these pumps, what you're doing is just, I'm making sure you can see it on camera. Yeah, you guys can see. All right, so we're going to squeeze out the air, we're going to put it on the tip, on the end of the pipette, and we're going to suck up the liquid.
Oh.
Now, it's going to take a while because I chose the 25 one, and hopefully there's enough air for it to take it. If not, then I just have to... I should have used a color liquid.
Oh, that's so cool.
So what you have to do, the problem with this one is that you don't have the same control. This one you have a little bit more control over because you can actually adjust the air pressure. So you'll find which one you like. I'm just gonna put this back on, suck up some more liquid. Now the thing with this is that you want to overshoot, so that way you have control to release it down to the line. Some things you need to keep in mind before you pipette anything, this is going to be in the side pad pages, is you want to get a paper towel and dry the outside bit to remove any excess, because that's going to be potentially added to your unknown. But first, before you even do that, you're going to release your hand, just a bit, so that way the suction breaks, and some of that liquid comes out. And then you stop it so the bottom of the meniscus is next to the line. Dry it off, and then you would put it into a flask, and this is known as an aliquot. So what this in here is the aliquot, and I release it up. So there's lots of different tools you can use. It's going to be some time to figure out which one you prefer. I'm just going to let it go through, and then I'm going to talk about what else you need to do as a finishing thing. I did 25 mils, so it's going to take a little while. Nice? Alright, now it's nearing the end and not everything's going to come out, and it is designed to factor that error in. What you do want to do is you want to touch the tip of the pipette against the flask so the water tension pulls out, and that way you get the accurate value. But there's going to be a little bit left over, and that's okay. It's designed to have that error in there. I don't want to see you guys grabbing your pumps and then trying to the extra drop in, because that's not what you're supposed to be doing. Okay? So yeah, those are the two main bits of equipment. I'm mindful we have just under 10 minutes left. I don't think it's enough time for you guys to take the equipment out and play with it and put it back. But when I see you guys next, we will use the 70 minutes for you guys to come out and test it out. There's some side pad pages you can go ahead and start on. So you can start just doing some of the practice, understanding of reading the meniscus, and understanding the techniques. There's also that online worksheet you guys want to use to try to make sure you know your vocabulary. Sound good? So you use this time to either do the side pad or you can do the online worksheet. Cool. And then when I see you guys next, you will be able to tinker with this with water. All right? Any questions? No? All right, hop to it. I'll put those in touch with my Alright, so we where I want you to start thinking about titration. So there's the online worksheet if you guys want to work on your device. There's 2 pages of the side pad you can do, which is what I've been assigning as homework. The first page, I just want you to do the first half of it, reading the meniscus. You won't do the second half because the second half you're going to do in class because you want to test out the BRF. Or you can work on page 13, which is using the pipette. 12, 13 are the 2 pages of the side pad I want you to work on. There's also
You want us to work on it?
you can work on it now. You got 5 minutes. Get it started. Don't waste time. Just always those 5 minutes add up. These 5 minutes that you don't do at the end of class, it adds up. And then you find yourself, you've done, you miss an hour of work. Is it on the line or is it so on the line with 0? Or would you like to 5? Does that make sense? Some of these, we look at the bottom. And so then you put Philadelphia, and then might be you could put 5 or 0. Yeah. So the ending number will be 0 or 5. 0 on the line, 5 if it's remember 2 decimal places. Does that make sense? Yeah, because you want to say either is it on the line or is it between 2 lines? Between the 2 lines and a 5. That makes you say it's half. 2 decimal places. Units as well. Oh my goodness. Is it milliliters? Yes. Gotta readjust your finger. Trust me, I've had so many kids that cheat because they read it all wrong. So remember going in the top. Yeah, so you're going to
I'm so fun.
You guys are picking this up very quickly. You're mistakes very fast. And don't have to last absent today, so you're gonna have to really do the rest.
Are we reading from the middle or is it?
I just
Like a rabbit.
You're writing it in the opposite direction. You're thinking about a measuring cylinder. You're thinking about these guys. Because you're used to. But you're measuring the water, you have to be mindful that the scale is in the opposite direction. Here, let me grab this for you.
I didn't even sleep well last night.
You gotta be quick.
My body was thinking. Wow. I have no idea.
What did they say?
I can hear it. Make sure you spray one on the tables. I'll let you have Monday to play with this.
Did you whisper?
Yeah. Or you can take them all. You're gonna leave them on that shelf that says level 2. Or in that box. Box works. Oi! Ruben! Hunter, turn around. You gotta spray and wipe down these tables. Ruben! Yes, Hunter, turn around. Spray and wipe down my tables. Excuse me. Spray over here. Spray and wipe down my tables. And return any textbooks that you borrowed. Thank you.
Shame. Everyone wants to know.
Appreciate it.
Remember that Sunday when you get really frustrated over like that this is really good. When you're sitting there, dang it, it's too big.
What did they do?
You gotta feel that way where the meme is on that checklist. Start going.
Boom.
That's the whole reason they call it titration. They'll tell you about how aggravating it is.
Thank you.
See ya, have a good weekend. You're welcome, have a good weekend.
Ooh.
Make sense?
Yeah. Bye.
See ya. You too. working with things.
Oh, yeah. You sometimes link psycho pages for the lines.
Yes.
Where do you find that? Do you have a book?
I think I have a digital copy.
Oh, okay. Because we're seeing a CD that could be far, but
Yeah. I don't see any on my shelf here. You need the deal. Well, actually, that's in your notebook. What I will do is I'll send you a digital copy of it. Not supposed to, but.
Okay.
It's a useful thing to have.
Yeah.
Just in my opinion, can't click from it. That's why I screenshot it.
Oh. Yeah.
I'll also go through our demo of sparklers as well at some point, and I'll send those to you, the sparkler worksheets for those literacy ones.
Oh, okay.
Very useful.
Is it? Okay.
So,
Yeah, this is great.
yeah.
This is fun.
You can ask around and see someone else has this iPad. You can find me.
Okay.
Yeah, I'll- Yeah, we're doing titration. I was introducing it. It's fun. I was like, this sounds so fun. Listen, keep that in mind when we start doing the titration and you get frustrated
Oh, yeah.
that you enjoyed it at one point. Because you're not going to enjoy it when you start. Oh, that was still running. Whoopsie. All right, so just to recap for today's lesson, we were looking at the purpose of quantitative analysis and the titration equipment. We probably definitely got the purpose covered, but we still need to work on the equipment. So next lesson will be focused on giving you guys some time just to tinker with things. But you should start getting an idea of what the vocabulary for this standard is.